Hello everyone and welcome to chapter 3, which is entitled Cellular Form and Function. Now this particular chapter has been divided into two separate lectures. So in our first part, we're going to go ahead and start exploring exactly what a cell is, what is the cell theory, what is the cell membrane all about, and we're also going to take a look at some of the transportation mechanisms that a cell can utilize in order to get nutrients in and waste products out. And then the second part of the lecture is going to take a look at tonicity, which is basically how the cell will interact with its environment. And we're also going to take a deeper look at some of the organelles that make our cells, eukaryotic cells, very specific, and also are able to divide up their labor. So things like what does the mitochondria contribute, the smooth ER, the rough ER. It's a nice little overview of what's going on as to the internal workings of the cell itself. Now, before we actually start lecturing, I do want to remind you that you do have a copy of this PowerPoint available for you on D2L. So as you are listening to the YouTube video, feel free to move at your own pace. And as you're taking your notes, you do have the PowerPoint available that you can download and scribble all over. I do just want to point out that I tend to add a few more pictures onto the recorded section, but the wording and the text will be pretty much the same. So then that way you can take some nice comprehensive notes as you start getting ready to, get ready for your homeworks or your exams and whatnot. I also want to go ahead and give you a quick reminder that if you have any questions, comments, or concerns, please feel free to email me at any time. I hope to see you at our Blackboard office hours. And also I've set up discussion sessions on D2L where you can go ahead and interact with your classmates as well as myself, all in an effort to get you to a comfort level on the material that is involved with our anatomy and physiology course. Now, in chapter 3, as I mentioned before, we're going to go ahead and take a look at the cells. And one thing that I particularly like about the Saladin book is that it is going to go ahead and start us really small by taking a look at our cells, and it's then going to build from there. So we're going to explore the tissues in the next chapter, and then we're going to eventually get over to the organ level. So we're going to build up on our knowledge, starting with what we like to call the fundamental unit of life, which is a cell. So let's go ahead and take a look at the slide that's currently on our screen, and it introduces us to what we like to call the cell theory. A theory in science is something that has been well researched and established, and we have lots of years of research on cells, primarily due to the discovery of the microscope and our ability to figure out its internal workings. So it turns out that after all these years of research, the cell theory has allowed us to establish three tenets, three merits that we always have to live by. The first one is that the cell is the fundamental unit of life. The second one is that all organisms are composed of at least one cell. Now, what I mean with that is that you can, for instance, have a very simplistic cell, like a little bacteria cell, or here we have a cyanobacteria, which is a special grouping of bacteria that's able to photosynthesize. Both of these organisms are considered a unicellular organism, meaning that they are just composed of one single cell. They do not have a nucleus, meaning they don't have a control center within the cell that houses their DNA. It's very simplistic in their setup, but yet a fully functioning living entity. We can also have organisms that have a more complex structure, which is then going to often be included in what we like to call a eukaryotic cell. A eukaryotic cell means that there is a nucleus or a control center within the inner workings of the cell, and this can be found in unicellular organisms as well as in multicellular organisms. So for instance, if you take a look at things like amoebas or paramecium, these are all examples of unicellular eukaryotic organisms. But then also you have examples such as different plant species. How about animals such as ourselves? And also when you take a look at things like fungi, algae, these are all examples of different multicellular organisms that have hundreds, thousands, if not millions of cells, all depending on the size of the living entity. So the first one was that the cell is the functioning unit of life. The second part is that all organisms are composed of at least one cell. And then the third part of the cell theory came along a little bit later on, and that one says that cells come from pre-existing cells. That's correct. So cells don't spontaneously appear. They have to come from pre-existing cells. And a very good example of that would be us, for instance. Where did we come about? Well, many years ago, there was a single cell with a tail, let's call that a sperm cell, thanks Dad, who found its way into the fallopian tubes and came across another cell. We're going to call that cell the oocyte, or the egg cell, thanks Mom, and those two cells merged together to create a zygote, which is what we call a fertilized egg, and out of that fertilized egg came you. So, sorry to tell you that, right? So all cells come from pre-existing cells. So make sure to thank your parents for that one. All right, so let's go ahead and take a look at the size of the cell. Why is it so important for a cell to stay nice and small? Our body is composed of millions of different cells, and each cell can have a very particular function within the body. But when we take a look at the structure of the cell, we're often going to see that the cell tends to stay nice and small. And it turns out that that all has to do with what we like to call the ratio of its surface area and its volume. So just think about this for a second. If the cell is the fundamental unit of life, that means that it's a living entity. And as with all living entities, it's going to have nutritional demands because it requires energy for its day-to-day activity and its maintenance, but also it's going to generate waste products. And these waste products are going to have to find a way to exit out of the cell to maintain its homeostasis, its internal work balance. So, when you take a look at a cell, and we can go ahead and take a look at figure 3.2, which is on our slide currently, we see that they've drawn two cubes for us. And these cubes are going to represent our cells. The little purple structure on the inside, in case you were wondering, that represents the nucleus. That is where we house our DNA. And what we see happening here is that there's some calculations at the bottom of the slide. And within the calculations, they went ahead and they figured out what the surface area was and what the volume of the cell was. Now, let me tell you why we care about these two values. Well, the first one is surface area. Surface area is going to take a look at the external membrane of the cell. So this is going to be kind of like its, it's going to be its plasma membrane. This is going to be the boundary that's going to separate the cell from the inside and the outside. So if I were to draw a very simple circle right here and tell you this is my cell, the red line that I just drew, this is going to represent my plasma membrane, and my plasma membrane is going to expose me to my surface area. How much area do I have to allow nutrients to come in and waste products to come out, okay? The second value that we're going to take a look is going to be called its volume. And I'm going to change my color here of my little clicker. The volume is going to be interested with its internal mechanism, what's happening on the inside of the cell. So that's going to take a look at what we like to call the cell's cytoplasm. That's where we're going to find our different organelles like our mitochondria, our nucleus, our smooth ER, our rough ER. If some of these sound a little foreign to you, no worry, we do have a section at the end of the chapter where we'll take a look at these organelles and get to know what they actually do. Either way, the internal workings of the cell, so everything that I've kind of colored in green that makes up its volume, that's going to determine exactly how large the cell is on the inside, which will then directly contribute to how much nutrients need to come in and how much waste products needs to go out. Okay? Now, take a look at the calculations of the large cell and the small cell. I guess we should probably start off with the small cell. So here's our nice little small cell. It's 10 micrometers by 10 micrometers, which is microscopic. And when we calculate it, you can see right here, we have our surface area and we have our volume value. Now, it's not important to memorize these numbers. We're just going to see what the trend is going to be when we go ahead from a small cell to a large cell. Meaning, are the numbers going to increase? Are they going to decrease? Are they going to be synchronized with their movement? Or is one going to move faster than the other? Next, go ahead and take a look at your large cell. And as you can see in the large cell, what they've done is they've doubled the size. They have gone from 10 micrometers to 20 micrometers on each side, which is still very small. And when we double the size, you can come right over here and you can see that once again we have a value for our surface area and we have a value for our volume. Now, as you can see, both of these numbers increased as you transition from a small cell to a large cell. So what does this all mean for us? Well, we're interested to see in exactly how much that these numbers increase. And if you now go ahead and take a look at your surface area for the small cell, compare that to the value of the surface area in the large cell, you're going to see that the surface area has increased by a factor of 4 by simply doubling the diameter of the cell. So we went from 600 to 2400. So that means that the large cell has 4 times as much surface area to allow products to come in and waste products to come out. Okay, sounds good. All right, now let's take a look at the volume. What's happening in the volume? The volume from the small cell to the large cell had an increase of 8. You can see it right here in the blue box. So that means by doubling the size of my cell, I increased my volume by 8-fold. So that means I need 8 times as much nutrients as well as 8 times as much waste products is going to be generating by me simply doubling the cell from 10 micrometers to 20 micrometers. Now you're going to notice that both of the values, surface area and volume, both of them increased. However, notice that based on the calculation, the volume increased drastically more than the surface area did. And what we see happening is that as you keep increasing the cell, this trend will continue on, meaning that the volume of the cell will increase significantly faster than the surface area of the cell. The reason this is important to point out is because as the cell gets larger and its volumes, areas, or its numbers start to skyrocket, that means that the cell has a higher need for nutrient and waste products. However, it can't keep up with the demand of the volume because the surface area is not growing or expanding as fast as its internal workings. So the solution to have a very effective cell that can keep up with its demand of nutrients and waste product is to keep the cell nice and small. Because when you keep it nice and small, what we see happening is that the surface area that the cell has is effective and enough to maintain its nutritional demand and waste expedition that's happening on the inside of the volume. Now, what if you really do need to have a larger cell? So for instance, neurons, the cells that we have in our brain and our spinal cord, part of our nervous system, we have neurons that literally run the entire length of your body, right? Because if someone stomps on your toe, you feel that because it sends a signal over the brain saying, ow, that really hurts. So if you need a cell that needs to be large, how do we solve this issue? Well, how about we make the cell large, but we make it nice and flat? By making the cell flat, we see that we minimize the amount of volume. So we keep that nice and low, and at the same time, because we're extending the cell, its surface area flattens and reaches out and thereby also increases. So that's one way that you can manipulate the shape of a cell by altering, but keeping still in mind that you still need plenty of surface area to keep up with the volume, especially if you're going to be increasing a cell. So I understand that was a little bit of a long-winded explanation, but the takeaway message from this slide is that the cell works best when it has plenty of surface area to meet its volume demands. So the best thing to do is to either keep the cell nice and small, or if you want to go ahead and increase the size of the cell, find a way that you can stretch out the surface area and minimize the volume on the inside. So one of those would be to, in essence, flatten the cell. Here is a illustrated example of a eukaryotic cell, and this is just kind of like one of those typical cells that you would come across. Part of what we're going to be doing in our class is that we are going to be exploring all the different types of cells that we have in the body and how they will work together to give us different tissues. So for instance, epithelium tissue, which is what we deal with when we take a look at the skin, or our different connective tissue, our muscle tissue, our nervous tissue. And when we look at all these different cell types, one thing that we're going to notice is that there are a few commonalities that we're always going to see. Their structure might be slightly different, but they're always going to have certain features. One of those features will be the presence of a cell membrane. And as I mentioned to you before, the cell membrane is going to be the border that separates the inside and the outside of the cell. Now, some of you might know it as the plasma membrane, which is completely correct. Some of it might know it as the cell membrane, which is my preference of calling it. But either way, that's going to be the border. We're also going to see that the internal workings of the cell, so what's happening on the inside, this is going to be part of its cytoplasm. And that is where we're going to be finding all these different organelles, all these different colored structures that we see floating around. Here in orange, we have our mitochondria. Here in purple, we have our nucleus, where we're housing our DNA. Because of the presence of the nucleus, that means that we are looking at a eukaryotic cell. In blue, we have our Golgi apparatus. My apologies, that's not the Golgi apparatus. That's the ER, the rough ER and the smooth ER, the endoplasmic reticulum. Here is the Golgi apparatus, also known as the Golgi complex. Beautiful. Part of what we'll do, as I mentioned to you before, is at the end of the chapter, we're going to go ahead and take a look at what each of them contributes to a cell. But before we get to that, I want to talk to you a little bit more about the plasma membrane. What is it made out of? And I also want to talk to you about the different ways that we can sort of extend the plasma membranes within a cell so that we can interact, we can increase the interaction with the internal and external environment of the different cells. So let's go ahead and skip over to our next slide and take a look at what exactly our plasma membrane is made out of. All right, so here we have a beautiful image from an electron microscope. We can see our nucleus, and right over here, this little border, this is going to be our plasma membrane. Now, when you're looking at this picture, you're going to notice that obviously the plasma membrane allows us to separate our external cellular fluid, that's the fluid that's on the outside, from what's happening on the inside, which is called our internal cellular fluid. So that's where we're going to find all our organelles. And also what you're going to see from the picture here is that notice how you have two layers in your plasma membrane. You have an upper layer right here, and you have a lower layer. So our plasma membrane is what we consider a bi-layer structure. It has two layers that stack on top of each other. Now, what exactly is this layer made out of? Well, it's mostly made out of phospholipids. Phospholipids are structures that are composed mostly of glycerol, fatty acid, and a little phosphate group. And what's going to allow us to do this is that it's going to allow us to create both a hydrophobic as well as a hydrophilic environment for the cell membrane. Now, go ahead and take a look at the illustration that I've inserted into our little slide. And on your right-hand side over here, you can see that a typical phospholipid that makes up a plasma membrane has a little head section, here's the little circle, and it has a little tail section. And the head section is going to be considered hydrophilic. Hydrophilic, hydros for water, philic is to love. That means that that particular section can freely interact with the water. Now, it's important because our body is mostly made out of water. So there will be lots of water that the cell will have to interact with, both externally and internal of the cell. The tail end, on the other hand, is going to be hydrophilic. hydrophobic, and that means that it's going to be water-fearing, so it repels the water, which sounds a little weird. Why would we have a hydrophobic component in our plasma membrane if we know that our body is mostly made out of water? But it turns out that we need both of these elements, the hydrophilic and the hydrophobic components, to create a semi-permeable environment. Semi-permeable means that the plasma membrane will be able to control what can come in and out of the cell. So it's fantastic of using phospholipids because it will allow it to create an area where it can freely interact with the water and also where it can repel the water, thereby once again creating its semi-permeability. Now, in case you were wondering how exactly does it stack on top of each other, you can see right here on the slide, it says the hydrophilic head will face the water on each side of the membrane. So that means both the outside external cellular fluid and the inside internal cellular fluid, while the hydrophobic tail will be folded inwards into the center. So come back to your picture and you can see right here that they're stacked, that the head that's hydrophilic is pointing to the fluid, whether it's pointing to the outside of the cell and it's pointing to the inside of the cell. And then right over here, you can see that the tails are folded away in the center, allowing them to be kind of protected from the water because remember, they do have that hydrophobic component. Now, keep in mind that us stacking our bilayer this way will give us our semi-permeability or selective permeability component to the plasma membrane. It will also allow the cell membrane to be very fluid. So your cell membrane is not a rigid structure, it's very fluid, and it can easily move from side to side in a lateral motion. Here is another illustration of the plasma membrane, and right over here in yellow, we can see the hydrophilic heads and the hydrophobic tails. You can see the bilayer, that you have your top layer right here and your bottom layer stacked on top of each other. And what this picture also shows us is that our plasma membrane, in addition to it being semi-permeable and being fluid, that it also has a mosaic model. Mosaic means that it's made out of different components. And what that really entails is that within the plasma membrane, within all those phospholipids, you will have lots of different proteins that will be embedded within it. And these proteins will have different functions. So here are just a few of them. For instance, some of them will allow us to allow the cell to adhere to each other, which will be very important for the cell not only to communicate with its neighboring cells, but will also be important, for instance, when we start taking a look at how we can use cells to build up tissue. We can use some of these protons to move ions. Excuse me. We can move these ions into and out of the cell, so things like sodium, potassium, chloride. We can use these membranes as identifying markers. In fact, all of us have little glycoproteins, which are little sticky antennas that stay on top of our cell membranes. You can see it drawn right here. And these allow our cells to identify which cells belong to us and which ones are foreign. So like a bacteria cell or like a virus-infected cell, very effective for your immune system. And all of us have very unique cell identifying markers, with the exception of identical twins. Because they share the same DNA, they will have identical markers as well. And as here on the left-hand side, you can see that there's also some receptors. The receptors are going to come in very handy, for instance, if you have things like chemical messengers. So think along the way of your hormones that are released into your bloodstream. And when they get to the target cell, how exactly do they communicate with that cell? Well, there will be different proteins there that will allow it to interact so that it can induce a reaction out of the cell that it's needed for. Now, along the surface of the plasma membrane, we also see that there will be certain mechanics or certain structures that will allow the membrane to extend itself, allowing it to increase its capability to interact with the environment. And this will often be handy, for instance, to allow for a wider range of nutrients to be detected and absorbed into the cell, and also obviously to get rid of waste products at a faster rate. One of these structures that are very helpful in increasing or extending the cell membrane's capability are what we call microvilli. Microvilli are tiny little hairs. I often like to tell students, think of them as little tiny little fuzzy baby hairs that sit on the outside surface of the membrane, and they create these little infoldings. You can see it on the little picture right here. And I'm doing my best to follow the infolds of the microvilli. And it turns out by making these little infolds, what we're doing is we're increasing the surface area. So we're allowing the cell membrane to extend way beyond its capacity, which will often increase its absorption rate. A really good example of where you would have microvilli would be, for instance, in your digestive tract. As food travels from your stomach into your small intestines, what we see happening is that when the cells become small enough that their nutrition is being released, we see that our small intestines is lined with microvilli, and they create what we call a brush border. And these brush borders allow the interaction of the nutrients on the inside of the small intestines to be then absorbed into the bloodstream on the outside of the digestive system, so it can quickly make its round throughout the rest of the body and distribute its nutritional value. So microvilli are these small microscopic hairs that are often bundled together. Another example of how we can extend the absorption rate or the surface area of the cell membrane is by taking a look at cilia. Cilia is going to be longer than microvilli, and it's often not as numerous. Cilia is often found in a lot of our orifices, so anything that's exposed to our environment. So for instance, our oral cavity, our nasal cavity, our reproductive tract, our urinary system, they will have lots and lots of cilia that will guard these openings. And part of what the cilia will do is that it will create these wave-like motions as the hairs swoop left to right, and they're able to catch any type of pathogen that might try to make its way in, and it will wrap that pathogen often in mucus to immobilize it. A really good example of that is, for instance, in our nose. We have little Fabricia, little nose hairs that are able to trap any type of pathogen. And when the pathogen enters through our nose, we obviously don't want it to get, for instance, to our lungs. So what the cilia will do is it will trap it, and then we wrap it around mucus, and that, of course, is what we like to call our boogers. That's what we're cleaning out of our nose. All right, now cilia doesn't always have to move, as you can see on your notes right here. We have some non-motile cilium. These can be used for anything like monitoring the environment, or for instance for sensory cells. And then in our orifices, we tend to have the more motile cilia so that they can track the pathogen, wrap it in the mucus, and of course escort it to another area, like maybe you're coughing, like I was just doing, my apologies, or if you sneeze, anything to kind of exit that pathogen out of the body. On this slide, we can explore the mobile cilia just a little bit more. Go ahead and take a look at the picture that's on the bottom right-hand section, and you're going to see how the cilia, represented by these long hair-like structures, are able to create what we call a power stroke and a recovery stroke. And as you can see, looking at the little black arrows, the power stroke and the recovery stroke almost create like this wave-like motion that the cilia will use to keep the mucus or any type of pathogen that it has trapped, to keep it in a certain location and then transport it over to a safer spot. So this could be, for instance, that it's moving it up towards the mouth where we start to cough and then eject it out of our system, or we clean it out of our nasal cavity, or even within the throat a little bit deeper in our anatomy, we have what we call a mucociliary escalator. Yes, an escalator. And that's because the cilia will move the trapped pathogen up the trachea so it can dump it into the esophagus, where that pathogen will then go over to the stomach, where its acidic environment will often kill it off, thereby doing no harm to the body. One thing that you can also see from the cilia is as it's moving, it allows the mucus to almost glide on top of the hairs. Now, something about the structure of mucus, if you were to describe it, I think many of us would say that it's very sticky. So how does the hair not interact with the mucus, right? Because that could kind of clog up the fine structure of the cilia. Well, your body has come up with an ingenious plan for that. It turns out that anytime when we take a look at cilia and its power stroke and its recovery stroke, oftentimes it will utilize chloride pumps to create a little salt or saline layer. And the saline layer will completely submerge the cilia, and it will allow the mucus to float on top of the saline layer. And then inside the layer, we can have the power stroking and the recovering stroking going on, creating the motion that's needed to move the mucus from point A to point B. In fact, we know the importance of these chloride pumps because they've been well studied in individuals with cystic fibrosis. Cystic fibrosis is a genetic disease where these chloride pumps are either absent or they're no longer in functioning. This then means that the mucus has free interaction with the cilia and it clogs up the hair. And when the mucus then clogs up the cilia, it's not able to move or eject itself. And mucus then becomes a harboring area for bacteria. That's one of the reasons why individuals with cystic fibrosis will often have extensive treatment therapies that they'll have to go, and they're very prone of getting any type of respiratory diseases like pneumonia, and that's because of the fact that their mucus has a very hard time moving from point A to point B due to the lack of the chloride pumps. Another movement that we can utilize to extend the plasma membrane is the flagella. Now, the flagella is the longest of the extension. And when it comes to the human bodies, gentlemen, I want you to be very proud because you are the owner of the only flagellated cell that humans have, and that's going to be your sperm cell. So I've inserted a little image right there of a sperm cell, and as you can see, its tail and its wave-like motion, that is the flagella. There are lots of different organisms that will have flagella. Bacteria tend to have multiple ones, and as I was mentioning to you before, in humans, the sperm cell is the only one that has it. And as you can see, it's excellent for locomotion, and it creates these whip-like structures. They are very similar in movement when we take a look at the cilia, but of course with the flagella, these are going to be much longer whip-like structure, and they're also more coarser, thicker than the cilia and the microvilli that we have mentioned thus far. And at the bottom right here, it just kind of tells you that it's more of a power stroke in the sense of that it does like a corkscrew motion. So it's not the clear backwards and forward motion that you saw in our previous slide when we were talking about the chloride pumps and the cilia layer. Another thing that I want to mention for plasma membranes and to kind of wrap up its ability to kind of increase its interaction with the environment is the use or the formation of what we call pseudopods. Pseudopods is when we see that the entirety of the plasma membrane, so not just some fine hairs like with the microvilli or the cilia or the flagella, but when the entire plasma membrane is able to have this fluidity and this ability to constantly change its extension. So we have a little illustration playing on our left-hand side. We could see a little, that's a little amoeba, and notice how its plasma membrane is able to create what we call little false feet. These are little extensions that will allow it to extend its interaction with the environment. We can also see it on the illustrated example that are on the right-hand side, where we can see how it's able to increase its surface area interaction. Here is some pseudopods interacting in what we like to call macrophagic activity, that they're basically swallowing or engulfing any type of foreign pathogen. Here's a little white blood cell doing its job, injecting the little bacteria. These are all cells that we're going to come across as we start exploring the body a little bit more. So this is what we call pseudopods, which are extensions of the entire plasma membrane. excellent for not only allowing for the fluidity, so it can often change the size and the shape of the cell that you are studying. All right, so now that we have become a little bit more familiar with the plasma membrane and the cell membrane, let's go ahead and take a look at membrane transport. This is going to focus on what exactly the cell uses to get stuff into and out of the cell. Now, this will then lead us back to our plasma membrane, obviously, and I want to remind you that based on our previous discussion, I mentioned to you that the plasma membrane, because of the bilayer of the phospholipids, it is selectively permeable, meaning that it can regulate what comes in and out of the cell. So, I'm going to draw a little cell for you on the side right here. So this is my little cell. And part of what I want to discuss with you is that when we take a look at membrane transport, you can either have what we like to call passive transport or active transport. It's all going to depend on the concentration that you're trying to move. Now, what do I mean with that? So let's go back to my little cell that I've drawn. And don't worry, there'll be a better illustration coming up on the next slide. And inside my cell, let's say I'm interested in studying salt. Salt is the most common ion that we have in our body. So I'm going to make little X's, and these are going to represent my salt molecules. So inside my cell, I'm going to draw 4, and then outside my cell, I'm going to draw 2. And what this represents is the fact that you have a high concentration of salt on the inside, and you have a low concentration of salt on the outside of the cell, right? More salt inside, less salt outside. Now, because you have the plasma membrane that separates these two environments, what we see happening over here is that we're going to be able to create what we call a concentration gradient. So we have two separate environments, inside and outside, and the concentration of our solute, our little particle, is going to vary. So we have a concentration gradient. This concentration gradient will then allow us to move either two ways. We can move from low to high, which means that we're going to move up the concentration gradient, or we can move from high to low, which means we're going down the concentration gradient. Oy. It's actually much harder to write down these words than you think. All right, there we go. Now, why do we care about all this? Well, let's come back to our notes. And it says right here, it says, if you have passive mechanism or passive transport, what we see happening here is that it will not require any chemical energy on the side of the cell. Whereas if you do active transport or active mechanism, it will require ATP, which is our chemical energy, thanks to the mitochondria. Well, it turns out that a lot of that, whether you're doing it passive or active, will depend on if you're moving up or down the concentration gradient. Now, envision you and a set of stairs. I hope you will agree with me that it's a lot easier to go down the stairs than it is to come up the stairs. Treat your concentration gradient the same exact way. It is easier for cells to go down the concentration gradient than it is to go up the concentration gradient. So when it goes down, when it goes from high to low, that's an example of passive transport. And this will not require any chemical energy because it will solely run on what we call kinetic energy. Kinetic energy is energy of motion. It's caused due to the fact that the molecules are constantly bombarding and hitting each other side to side. And this will then allow the molecules to go from a high to a low concentration. If you want to go up the concentration gradient, so you want to go from an area that has a low concentration to a high concentration, that's going to take effort, just like you were climbing up the stairs. So that's going to be an example of active transport. Active transport will always require ATP or chemical energy on the side of the cell. So passive transport, we go down the concentration, we go from high to low. It doesn't require any ATP. The driving force is going to be kinetic energy. Active transport means that we're going against the flow. So we're going to go from low to high concentration. We're going up the concentration gradient. And just like climbing the stairs, it's going to take extra effort in the form of ATP. As you can imagine, it's a lot easier for your body to do passive transport than active transport. However, it will utilize both, obviously, depending on what it's trying to move from point A to point B. All right, so here's a quick little illustration that I took from McGraw-Hill, and it's just to kind of show you involving our plasma membrane. We can see our plasma membrane right over here. You can see our bilayer. And as you can see, we have an example of passive transport here on the left, where we have our ions, whether it's in purple or the little red triangles or the blue little circles. You can see that you have a high concentration in one, and you have a low concentration, in this case on the inside of the cell, right? More on the outside than on the inside. Now follow the arrows and notice how the arrows are all going down the concentration gradient. So they're going from high to low. As you can see, some of them can move by themselves. Some of them will use what we call facilitated diffusion, meaning that they'll use some proteins to kind of guide them. Think of them kind of like a bridge or a door to help them move through. But either way, they go from high to low, and they require no ATP because their driving force is going to be kinetic energy. On the other hand, if we come over to our right-hand side, we now have a scenario where we are trying to move items from a low to a high concentration. And as we're moving it from low to high, notice how this is going to take effort. Follow the little arrow right here. And that is then why they have involved ATP. Anytime you see the mention of ATP or you see an illustration where ATP is indicated, please note that it will mean that you're doing active transport. So you're going up the concentration gradient, and this will include having additional chemical energy to get the work done. Now, I want to spend a little bit more time to talk about passive transport because we want to talk about two of the main examples, which are going to be diffusion and osmosis. All right, so let's start off with diffusion. Anytime you hear diffusion, I want you to think about two things. The first thing I want you to think about is that it's an example of passive transport. So that means that you're going to go from high to low concentration. The second thing I want you to think about is that diffusion means that you are moving solutes. You're moving little particles. So for instance, you're moving things like oxygen, carbon dioxide molecules. How about salt? How about chloride? How about calcium? How about glucose? Any type of molecule that you're moving from a high concentration to a low concentration, this is going to be an example of diffusion, which is an example of passive transport. The little illustration that I have playing on your right-hand side shows you a cluster of little red balls in the beginning, and notice how they'll disperse, covering the entire black square. And as they disperse, what we're seeing is an example of diffusing from high to low concentration. This will be an example of diffusion. We have lots of examples of diffusion in our day-to-day life. We could start with things like, for instance, if you have diffusers that are plugged in, or air fresheners that you utilize, or what about perfume or cologne, any type of splash on that you put on yourself to make you smell better? Well, the reason that others can smell you as well is because those little particles go from high to low, so they diffuse into our environment so that we can interact with that smell as well. How about if you love to enjoy a cup of tea, right? So we'll get a nice little cup of warm water, we'll put our little tea bag in it, and within no time we see that the water will often turn a different shade, and we'll start smelling the tea particle, we'll start tasting it, and that's because the tea particles have diffused. They have gone from a high concentration, which was inside the tea bag, to a low concentration, which was the surrounding water that the tea bag was diffusing into it. So please keep in mind, anytime you hear diffusion, that means that you are moving particles or solutes. They are moving from a high to a low concentration. It does not require any ATP. It will use kinetic energy as its driving force. And just a little FYI, you also have what we call facilitated diffusion, which is what you saw on the illustration previously. Facilitated diffusion is going to have the same exact concepts, but it often means that you're using a little protein as like a little channel or a little bridge to move the concentration from high to low. The other one I want to discuss with you is going to be osmosis. Osmosis is also an example of passive transport, which means that you will also move from a high concentration to a low concentration. But what makes osmosis different from diffusion is what you're moving. Osmosis means that you're only moving water. So the movement of water from high to low is called osmosis. This is going to be extremely important in our next little lecture series because remember, our body is mostly made out of water. So there will come a time where we have to discuss the fact that sometimes in an environment where you have an interaction of water molecules and solutes, so little particles, so chloride, sodium, potassium, glucose, there might be an environment where the water can move through the membrane, indicated by this little dotted line, but the solute is not able to move. It's not able to fit through it. So when you see that the water is moving, and you see that it's moving from a high to a low concentration, we call that osmosis. And once again, osmosis is an example of passive transport. It does not require ATP. It will rely once again on kinetic energy, and it will generate what we like to call osmotic pressure, which will allow the water to flow from a high to a low concentration. You can take a look at the illustration that's on the right-hand side, and you can see you have site A and site B of the little beaker. You have your semi-permeable membrane, and if you take a look, they've drawn the water as indicated by little purple circles. And then over here, the larger red structures, those are going to be your solute. Notice how the water can move freely, but the solute cannot pass through the semi-permeable membrane. Now the water on site A, you can see that there are a lot more water indicated by the purple structures versus site B. So if you set up an environment where the water can move through the membrane, we're going to see that the water will go from high, where it's most concentrated, to low. See this big arrow right here? So that shows you that the overall net movement is that it's going to go from high to low. And when you see this happening, you're going to tell me that this is an example of passive transport. You're correct. But more importantly, you're going to tell me this is an example of osmosis, the movement of water. Extremely important to understand this because we're going to talk a little bit about tonicity in our next lecture. We're also going to talk about the fact that osmosis will help us regulate how much water we hold on to. And if something goes wrong, we can have things like diarrhea or constipation that comes up. We also see that our kidneys, for instance, will have access to what we call aquaporins, which are little channel proteins that are specific for the distribution of water. And when the kidneys have to release extra water because we've been well hydrating ourselves, we see that they're able to generate or increase their aquaporins, get more channel proteins in there so that the water can move faster. And when we're not drinking enough, it will take those porins away so it can hold on to more water on the inside of the body. Thereby, we're urinating less. Alrighty, so I am going to stop part 1 of chapter 3 right over here. Whenever you're ready, go ahead and transition to part 2, where we'll continue our conversation and we're going to take a look at tonicity as well as the different cell organelles that we find in a eukaryotic cell. As always, please take your time as you're moving through the lectures. I also want to recommend that you take your notes, and if you have any questions, comments, or concerns, please feel free to email me. All right, so we will talk soon again in part 2.